Hi everyone, I'm Elena and I'm a PhD student in neuroscience in the University of Verona under the supervision of Dr. Chiamazzi. Today I'm going to present you our work on the study of how the visual conscious perception works. We work in the field of cognitive neuroscience, which studies the functioning mechanisms of cognitive processing, trying to understand how they can be implemented from a neural point of view. It is important to highlight that cognitive abilities that emerge are bound to the biological structure of our brain. Some of them are perception, memory, language, or attention, and we investigate them through different techniques. Some of them allow us to record the brain activity, such as the electroencephalogram. Others allow a structural analysis of the brain, such as the magnetic resonance, or even we can perturbate neural functions with, for example, the transcranial magnetic stimulation. We study the visual perception, so let's see an example of what we know about its functioning. We see a cat coming out from behind the couch. The light coming from the cat is converted in neural signal in the eye, and through subcortical pathways the signal arrives in the visual cortex. Here the information is divided in the ventral stream that is responsible for the object recognition, like in our case it is a striped cat, and the dorsal stream that is involved in the vision for action, like converging visuomotor information to pet the cat. At the moment, there is a discussion about the possibility of an involvement of the dorsal stream in the object recognition, specifically regards the shape recognition. So, in our project, we want to disentangle the functional contribution of the dorsal stream to object recognition, specifically in the shape perception. To do so, we use the transcranial magnetic stimulation. We stimulate two cortical areas of the brain to enhance the connection of the dorsal stream and to improve its function. And then we verify it by testing its effects on the ability in discriminating shape and features of objects. The logic behind this procedure is based on the Hebbian principle, which claims that when a cell A excites a cell B and repeatedly takes part in firing it, some metabolic changes take place such that cell A becomes more efficient in making cell B fire. So, in the same way, we stimulate two cortical areas in sequence to enhance their connection in a specific direction. In our case, they are the start and the end of the dorsal stream. What we predict from this experiment is that if the dorsal stream sustains the object shape recognition, we expect to find an amelioration in the discrimination of global shape. If it is not involved in the object recognition, we expect no changes in both global shape and feature discrimination. But if we find an amelioration in both global shape and feature discrimination, well, we will be very surprised. That could mean that the dorsal stream sustains both shape and feature representation, or the reason for these results could be that we induced a more generic arousal effect. In each case, our study contributes giving clues for existing contrasting data, shedding light on the role of the ventral and the dorsal stream in the visual processing. And in a wider view, it takes part in trying to disentangle the functioning of the visual system. These progresses will be relevant also for the clinical application because a more complete knowledge of the visual system allows a better cognitive profiling of different pathologies, a better support during brain surgery in preserving cognitive functions, and a better planning of cognitive rehabilitation. And in the future, the aim is to implement visual prostheses also for the visual cortex. Thanks for your attention and thanks to all the Panda Lab. Bye!